live with the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh. It's Science Tonight. Now here's your host, Chris Smith.
Good evening, everybody. How are folks doing tonight? Welcome to Science Tonight. I am your host, Chris Smith. This show brought to you by the North Carolina Museum of Natural Sciences in Raleigh, North Carolina. Thanks for being with us this fabulous Thursday night. We've got a great show for you this evening. We're going to be talking about a very important topic and one that in our present times is also very salient. We're going to be talking about vaccines. Tonight specifically, we'll be talking about a vaccine for HIV. Of course, vaccines are all in the news right now. And I feel like we've probably all learned a lot in the last year about how vaccines are made. But of course, they're not all made the same way, even though the process might be similar. So for tonight, we're going to be talking about ending HIV by developing a vaccine. We've got a very special guest. I want to remind everybody who's tuning in, if this is your first time watching Science Tonight, welcome. Glad that you're here. We're here every Thursday night bringing you great topics across the fields of science, technology, engineering, mathematics, even sometimes art and education and more. We talk to interesting people who are making discoveries every day in their fields of study. And we get to learn something new pretty much every Thursday, which is my favorite part. But of course, one of the great things about the program is that we also get to learn what you're interested in. Because every show, we do an audience interaction segment. So if you're watching on YouTube or over on Facebook, make sure that you are in the chat box and in the comments as we go, because I've got those pulled up. They're ready to go. Moderators are standing by to take your questions, get them to me so that I can ask your questions live on air of our special guest. So as we go throughout the program, make sure that you're in the chat box, letting me know what you're thinking about the program as we go. Interaction is key. I want everybody to participate. Of course, you can also just jump in the chat and drop a little waving hands emoji and say, "Hi, Chris, good to see you. Great to be here," because I hope that it is. Now, you can also, folks, maybe not right now, but at the end of the show, visit naturalsciences.org, the museum's website, and make sure that you check out all of the virtual events that we have going on. There are events for all ages happening just about every day of the week that you can participate in, sign up for. So great learning happening, but also lots of great fun stuff out of the Museum of Natural Sciences. Okay. That was, I think that was all the housekeeping. That was all the announcements. Let me bring on tonight's special guest, everybody. I'm excited to introduce this person to you, Dr. Christine Daniels. Dr. Daniels is a postdoctoral researcher at the Duke Human Vaccine Institute at Duke University in Durham, North Carolina. Dr. Daniels has research experience in cancer, immunology, molecular biology, stem cell biology. So I'm very excited that we've got an expert with us tonight on all of these fields. This is going to be a great show. But importantly for tonight's topic, Dr. Daniels has been working on the design, production, and evaluation of a vaccine against HIV. Christine, welcome to the show.
Thank you for having me. It's great to be here.
So, yeah, I'm so glad that you could join me tonight. I think maybe my first email to you, I mentioned where I came across your research, but it was actually on Twitter. And I think it was some of the first couple of posts that you made. And then the Duke Postdoctoral Association was this person is great. We want to tell her story, and shared information about you. And I was like, this is great. Look at this awesome research. Let's get Christine on the show. So everybody go follow Christine on Twitter. That was that whole bit.
Appreciate it.
So we'll be talking about wait, okay, let's just do this at the top. Tell everybody where they can follow you on Twitter, and then they can be scrolling while the show gets rolling here.
Oh gosh. Okay, you can follow me on Twitter at immuno. So immuno like immunology underscore IMMUNO underscore YANKNO. Immuno. There might be a W at the end. I don't tweet a lot, so I can't remember.
See, smart people always come up with the best Twitter handles. Mine is like just my name, basically. But smart people come up with great. Well, we're going to be talking all about your research, but I'd like to get to know you a little bit better and help our audience get to know you a little bit better as well. So right here at the top, I've got a few, like just easy get to know you questions that I hope you're up for. I've got just 4 questions for you. And folks watching online, drop your answers to these questions in the chat box as well. I'm very curious to see what you all think about these questions. So, Dr. Daniels, are you ready?
Ready? I'm ready.
Okay.
The sound's cutting off.
First question for you. Oh, okay. Sound's cutting in and out?
Yeah.
We'll see if we can work with that.
Okay.
Thank you.
Not a problem. The Zoom era.
The Zoom era, right? Okay, first question. Is a picture worth 1,000 words?
I guess it would depend on the picture. I mean, I've been to museums where you see these big canvases and there's nothing but like scribble on them, like a straight line or something like that. So I think it depends on the picture. I don't think I could come up with 1,000 words to say about some of them, ones I've seen, but yeah, certainly the picture.
Depends on the picture. Okay. All right. That's a great answer. Getting to know you already. Okay. Number 2, does pineapple belong on pizza?
I'm almost offended. I would be offended. I used to be a non-believer, but one of my friends converted me. They made barbecue chicken pizza with barbecued pineapple on top. And I don't know if it was the barbecuing that made it more palatable, but that one time it was okay. Other times I'm not so sure, but that one time I'll let it slide. So
if it's your friend's barbecue chicken pineapple pizza, thumbs up. If it's not your friend's barbecue chicken pineapple pizza, you're like absolutely not a fan.
Not my favorite.
Now see, okay, there we do disagree. There we do disagree. I can put pineapple on any pizza, anytime, anywhere.
I'd probably just eat it separately. I'd probably pick off the little pineapple pieces and just eat them and then eat the pizza. So I won't, the pizza won't be wasted. It's just not the best use of pineapple.
You're not going to just dump the pizza. Okay, well that's good to know.
Yeah.
We could find a way to share pizza. Okay, next one that I've got for you. What celebrity do you get mistaken for?
Oh, wow. I don't think I look like anybody. I don't think I have a doppelganger, but if I could choose, I would say Gabrielle Union, but that hasn't happened yet, but it may in the future.
There you go. Yeah. We can put that one into the it's on the internet now. So it exists in the universe that you can be mistaken for Gabrielle Union. Maybe. I'm glad we accomplished one thing tonight. Okay, and lastly, when we talked before, you mentioned that you were from Atlanta, Georgia, right?
Very proud.
So in that case, CocaCola or Cheerwine?
You're gonna make me get disowned.
Use your words carefully.
Yeah, don't disown me, my city, my state, but Pepsi.
Did I have that as an option here? Pepsi.
Pepsi. I'm
Well,
not a fan
okay.
of either one.
Okay.
Yeah.
You know what? I was thinking Cheerwine as a North Carolina staple up against an Atlanta staple, Coca-Cola, but Pepsi fits the same bill actually. Pepsi
Yes, a
is
cola.
from New Bern, North Carolina, and
Really?
it's from North Carolina too.
Gosh.
Absolutely, yeah. Pepsi was first made in New Bern. So, okay, you know what?
I
Everybody
thought you
can
would drink.
drop a round of applause.
I was gonna
I'm
say
sorry, say that
Cheerwine.
again.
Actually, I probably would have picked Cheerwine. So I guess North Carolina has a good reputation with drinks because it's not a fan of Coke.
Not a fan of Coke. All right. Well, let's see. Let me tally up the points here. And all those were exactly correct answers. So congratulations. We'll put you in the Science Tonight Hall of Fame with an A+ on the Get to Know You questionnaire quiz tonight.
Never won anything. Thank
Round of
you.
applause.
I'd like to thank my mom.
How many minutes do you need for your acceptance speech? And
I mean, this isn't something I rehearse in my spare time or anything, but I could probably extemporaneously think of one. I'd like to thank my mom, my family, the institutions and people that have supported me along the way in this journey to get to this moment right here. I'm just so incredibly grateful. I don't have the words to express how much of an honor this is. So thank you.
that's brilliant. In fact, I can imagine as you're being hooded at Duke University and they're giving you your PhD, it probably equated with that honor. No, it didn't.
Oh,
Not at
definitely.
all. We won't even pretend.
Top 2 and not number 2.
Christine, let's talk a little bit about your science. Let's get into the real reason that you're here and the real reason that everybody who's tuning in tonight is here. We want to learn a little bit about you, your research, and what developing a vaccine for HIV means. So give us the sort of elevator pitch for you, your research, and what you're doing at Duke.
All right. So as you kind of alluded to, I'm working in the field of HIV vaccine development. And what I'm on a daily basis, I'm trying to develop a strategy to come up with a vaccine that will provide protection against multiple strains of HIV. And to do this, our lab and our institute uses a reverse engineering approach. And what that entails is instead of focusing on starting from the virus as a point of vaccine design, we actually start by studying individuals who have been infected with HIV, but have not gotten severely ill. So we study the responses that they develop as a way to try to learn more about how to design a vaccine. And so by studying those individuals who have HIV but haven't progressed to severe illness, we know that we've been able to determine that they have antibodies that can bind the virus. And since no one has actually ever fully recovered from HIV, they're the best model we have for what a productive immune response to HIV should look like. So we study those interactions, we isolate antibodies from those individuals, and we study how their antibodies interact with the real virus. And by studying like that lock and key mechanism of how the antibodies recognize the virus, we're able to design replicas of the virus or how the antibody would engage it, or even replicas of that portion of the virus that the antibody binds. So similar to how we use the spike protein as a vaccine for COVID-19 or for SARS, we do the same thing for HIV. And so we design those replicas, we screen them in the lab doing a lot of tests and assays, and that's how we determine our candidates. And then we put those in the animal models to evaluate how well they actually elicit antibodies that can neutralize HIV. And so in a nutshell, that's what I do.
You said it in a nutshell, but at once it sounded simple and very complicated.
It's very complicated. Yeah, for sure.
So when you're in your research, how did you begin working on HIV? Like when it comes to developing vaccines, is that where you've been interested in doing research? Is it HIV? How did you come to this problem specifically?
Great question. Actually, no. So HIV was just like a happenstance occurrence. I'm genuinely interested in studying anything. I'm interested in reducing health disparities. So any disease that disproportionately impacts people of color has a special place in my heart. And so my background, that's why it's so varied, because I've typically done cancer research, but I generally believe that we shouldn't. So I'm interested in using immune-based approaches to to curing diseases. And so when it comes to HIV or just solving, sorry, I apologize. Sorry. I'm a firm believer that we shouldn't reinvent the wheel. And so as scientists, we tend to hyper-focus, because we're scientists, on all the cool things we can do in labs. So we think about what we can build and all these tools and technologies we have and try and design the perfect molecule, when in actuality, our bodies are actually really great at discerning self from non-self. And so rather than us spending millions of dollars or just years trying to recreate this, I think it's best, or I'm really interested in using our immune systems and leveraging this already existing platform to target to fight disease, basically, or enhance the ability to fight disease when we're able to. So I landed in HIV because, as I said, my background is very varied. I majored in anthropology, I did cancer, stem cell, all these different types of science, and I realized that I wanted to do immune-based therapies. And so I chose HIV because at Duke, there's a vaccine institute, and it's basically this consortium of different scientists who are highly specialized in different areas, but are approaching the same problem from different areas. And so for me, as a person coming as an outsider, I thought it would be easier to learn or come up to speed really quickly in an immersive setting where I have all of these experts in one place, rather than working in a place where I learn from one person and I get one perspective. I can get a much broader training and a really thorough training. So I chose the institute, and the lab I chose happens to work on HIV. So HIV kind of fit nicely in the picture, but it wasn't necessarily an intentional choice to pursue.
Okay, I see. Well, very interesting then that you found yourself working on this issue now and become so knowledgeable about it. And now you're working on trying to find a way to essentially help people with HIV, or help people who could, help everybody, I suppose, right? Help everybody. Can you, since we're still kind of early in the show, can you give us a sense of what HIV is, or at least the way that you as a research scientist thinking about HIV, like folks I feel like are pretty well versed in maybe, HIV is the virus that causes AIDS, and, some of the means of infection as a sexually transmitted disease, but, and other ways of being infected. But what's your perspective on HIV, and how does that inform the work that you're doing?
So kind of piggybacking off my last answer, like as an individual, for me, HIV is a tool for me to learn how to better understand our immune system and how we can elicit it how we can elicit very specific responses using the tools at our disposal. So for me, HIV is a tool to learn about immunity, immunology, vaccine design, how you can trigger certain parts of the immune system to react and cause them to evolve in different ways. That's the way I think about it. But certainly it is a virus that leads to AIDS in individuals that's sexually transmitted or transmitted vertically from mother to child.
You know what just popped in my head? Excuse me, forgot to mention that for all of our viewers, Dr. Daniels is here representing Dr. Christine Daniels and not the Duke Human Vaccine Institute. My apologies, Christine, that I missed that at the top of the show. That's important. The lawyers just breathed a deep sigh of relief. I felt it. Now that that's out of the way. So tell me a little bit about the process then. How are you going about studying HIV and creating a vaccine?
So conceptually, it's pretty simple. In actuality, it's pretty difficult. But we study those interactions, as I said, between the host. from the people who have been infected with HIV and the virus itself. And we use the structures of those interactions to design novel vaccine candidates. And so we call these recombinant proteins, similar to the spike protein that that's used to elicit an immune system for the COVID vaccine. So we use the envelope, which is the HIV spike. And so we make those recombinant or fake versions of the protein, and we can introduce modifications in that can help the antibodies recognize it more easily. So sometimes that means deleting a region or treating it with different drugs to make it more visible to the immune system. And so we experiment with different modifications of the viral envelope to make it more, as we say, immunogenic, so your body has a better chance of recognizing it and generating a response. So I actually do that part of the work. I help design some of the immunogens or vaccine candidates, and then I test them in the lab. So I'll look at things like how well they're able to bind to antibodies that we know recognize the real virus. That's an indicator that what we've made in the lab still looks like the natural virus, because we don't want to train the body to recognize something that's irrelevant. So I'll do lots of different assays just to kind of characterize what I've designed to see how well what we've designed in the lab matches the naturally occurring virus. And then once we think we have something that's very promising and that's recognizable by the immune system in vitro, then we'll put it into different animal models and see how well we're able to elicit specific antibody responses. And so we'll vaccinate animals and we'll characterize their responses over time and see, and we'll determine like what sites are being bound, how well those antibodies are able to engage HIV envelope, can it neutralize HIV envelope, how they bind, different things like that. And from that information, it's a very iterative process. And so what we learn from one study we'll use to create changes in the next immunogen to make it better. And so progressively, we move closer and closer towards getting an immunogen or a vaccine that can elicit the response that will actually provide protection.
Okay, so, in this process, like I said earlier, I feel like a lot of people have, at least folks paying attention to the news and other social media, even we've learned a lot about vaccine production and a little bit about the research that goes into them, how they're made, some of the different types of vaccines, even like folks are at a different knowledge plane now when it comes to vaccines than they used to be. Have we learned anything through this process of developing a COVID vaccine that you feel like you can use to develop an HIV vaccine?
I mean, certainly COVID is different in that we've had way more people to test things in really quickly. So when it came time to getting the drug approved or getting the vaccine approved, it was just really easy to get the numbers needed to study the effects of it. I guess I would say COVID has definitely served as a proof of concept since the COVID vaccines are devised on a similar strategy from what we use for HIV and actually informed by the strategies we use. So since HIV has been a 30-year plus problem that we've been trying to tackle, it's nice to see that the approach we're using isn't amiss, that it can actually work in a setting with a virus that's more amenable to this platform. So it gives us hope that, we're on the right track for HIV, and we just need to keep tailoring our immunogens to get the response that will be required for neutralization. So it's hopeful, definitely.
Okay, well, that's interesting. But like you said, we've been vaccines and cures for HIV for 30 or more years now, right? Like you said. So, but we got a COVID vaccine in 12, 13 months. What's the difference there? Is it just that we hit global pandemic with COVID and we just rushed it through and there's not that kind of urgency for HIV? Or is it actually something about the process of the virus?
Great. That's a great question. And I would say that it's a little of both. The urgency, the global-wide pandemic definitely created a sense of urgency that fostered collaboration in ways for science particularly that we've never seen. I mean, the sequence was made readily available in January. And that was shared between institutions, whereas in science, that's typically not how it operates. Everyone has their niche area, and we don't really share too much of what we're working on while we're working on until it's published, because there's a risk of getting what we refer to as scooped, or somebody publishing your research before you're able to and getting credit. And so for COVID, there's been a lot of those rules have been, those barriers have been knocked down, so that way we can facilitate faster development of a vaccine. So that was, phenomenal. I don't think we've seen anything like that before, as far as all of the different entities that have made concessions in order to work towards a common goal. And I think that that's fantastic. And I think, if anything, I hope it's the example and not the outlier. As far as, I forgot the other part of the question. Sorry. The actual virus itself is has differences that make it more challenging than SARS-2. So HIV, particular, it's very genetically variable and mutates at a much higher rate than SARS-CoV or even flu. So if you think about flu mutating on like an annual cycle, so you have to get a new vaccine every year. HIV mutates not only every time it replicates, but it can mutate fast enough that within a single individual, you can isolate different variants. And so like it has the ability to recombine with other strains. And it's just really genetically diverse. So if you think about when you're trying to design a vaccine, with COVID, for instance, we have vaccines that we've seen the emergence of about 3 or 4 new variants, and we're worried that those vaccines can't address or provide protection against those variants. Now think about if those variants are occurring in every individual that's infected in real time, how much more difficult it would be to design a vaccine that could provide protection. So the genetic variability is a huge challenge, and there are different approaches people have taken to try to address that. There's also the nature of HIV. So it's covered by these glycans or sugars, which a lot of viruses are, but HIV is one of the most heavily glycosylated proteins or viruses. And so those sugars actually block a lot of the sites that would normally be bound by neutralizing antibodies. And so, if you think about virus neutralization like a lock and a key, the key has to be able to fit into the hole, or it has to be able to recognize that site. And if it's blocked, then that prevents the ability of the virus to be neutralized. So when we're designing vaccines, we have to find a way to elicit antibodies that combine to not only the virus itself, but those sugars. And that's really challenging because a lot of those sugars look like sugars that are also in our bodies too. So if we design antibodies vaccines that elicit antibodies against those glycans on the outside, then they could potentially react with glycans that are in our bodies and cause an autoimmune response that could be really harmful. So there's a very specific type of antibody that's able to neutralize the virus in a way that won't react with our bodies. And that type of antibody is just really difficult to elicit, not only because of the specificity and needing to be able to bind the virus and the sugars, but also because in order for it to get that ability, it has to be really mutated. So from an evolutionary standpoint, it has to go through multiple iterations of evolution. I guess it has to acquiring mutations each time it replicates in order to get that ability. And so it takes a long time. So I guess to make it a more real world applicable, people infected with HIV can naturally develop antibodies that can neutralize HIV. We've seen that, but it takes years and years to happen. And since the virus is evolving so fast, it typically outpaces how long it takes an individual to develop those antibodies. So with vaccination, we're actually trying to cut that evolutionary process and make it shorter so people develop those antibodies quicker. So HIV is just a really, really complex virus. Also, it integrates into the genome, so it can form these latent populations that aren't targeted by therapies. HIV is difficult. This is not a 30+ year problem because for lack of effort. It's just really challenging, and that's kind of some of the draw for me. I definitely know I have something to work on for a while.
Yeah. Wow. That we know so much about it, that you know so much about it, to be able to think about the very specific solutions, right? Like how HIV works. Like we've learned so much about it, but it keeps changing. And so there's always something new that we have to investigate, always trying to learn more about it. It just sounds like a constant battle versus something like influenza or even SARS-CoV-2 COVID to try to fight when the enemy is always changing?
Absolutely. Yeah. So typically we try to focus on those sites that are conserved, but it's hard.
Tricky stuff. So, one of the things when I think about HIV and that I've learned recently in trying to read up about it and get familiar with it, to prepare for it tonight, is that HIV, though there's no cure for it, plenty of folks now live perfectly healthy lives because the therapies and treatments and medications that have been developed have given HIV positive individuals the ability to do so. Like, we have treatments and therapies, but they have to be on these treatments and medications. It's like daily medications in order to, you might have to jump in here to educate me more on it, but
I think
is a vaccine like a solution to that, or is it in addition to that? How does the vaccine fit into the current treatments and therapies that we have for HIV? That's the question.
Well, Chris, I think you hit the nail on the head. It's not, we do have quite a lot of therapeutics and modalities at our disposal for making HIV a very manageable disease. And people do live long, productive lives and are able to resume, some degree of normalcy. And I think that's fantastic. That's great strides for science. But even with all of those things, such as PrEP or antiretroviral therapies in existence, there's still about 2 million new cases of HIV diagnosed each year. And so what that says is that those modalities in themselves are not enough to fully lead to eradication of HIV. Further, when of those 2 million new cases, I think they estimate about 1 in 7 of those individuals isn't even aware that they're infected with HIV when they're diagnosed. And so that means that not only are they infected, but they're continuing to spread it. And so that shows us that a treatment standpoint will never lead to the eradication of HIV. So we absolutely need, I would argue that we absolutely need a vaccine to curb the spread and eradicate HIV. Yeah. Yeah, sorry. And the
so
lifelong
folks, if
part,
you're just tuning in to...
Sorry, the lifelong part.
No, go ahead. No.
I'm so sorry. I forgot the second part of your
question.
The
lifelong part.
Lifelong part. I think that in itself, from a Western standpoint, taking a medicine every day for the rest of your life doesn't sound like such a huge deal. But for the populations that are typically affected by HIV or the individuals, that's not always real feasible. It could be cost prohibitive. It could be an access issue. There's a lot of stigma. And also just the compliance issue, right? Like having to take something every single day without missing, or the consequences like maybe death. I don't think that that's a solution to me. I think it works for now, but I don't think that that's the finish line.
I like that. That's not the finish line. It's much better to find a way to get rid of it than to ask people to have to manage it every single day of their life. Especially, there are so many things that we ask people to do to manage on a daily basis anyway, right? Trying to keep track of a handful of medications, even though life-saving, that's a lot to keep up with. People are busy.
Yeah, people have other problems. When you think about the parts of the world that are impacted, a lot of these people are impoverished and live in resource poor areas. So, if I don't know if I have access to food, I'm not going to prioritize taking a pill. Or, if I don't have a home, or, there's so many other factors that impact someone's ability to take those medications. And, it's great that they're available when they are available, but I just don't think that it's a feasible solution to target the people who are like the most severely impacted by this.
Excellent stuff. So since we're a little more than halfway through, I'll remind everybody, if you're just tuning in, we're talking about developing a vaccine against HIV with Dr. Christine Daniels, a research scientist at the Duke Human Vaccine Institute at Duke University. If you've watched the show, if you're tuning in, drop your questions, your thoughts and comments into the chat and into the comments, whether you're watching on YouTube or Facebook. Here in just a few more minutes, I'll be turning the show over to all of you in order to see what questions you've got. And I'll be asking Dr. Daniels your questions. So go ahead and get those typing fingers going and get your questions into the chat so we can get them into the queue. So Christine, just before that little break, you brought up a great point, which is that there's stigma surrounding this. The highest prevalence of cases for HIV are in resource poor or in marginalized communities globally, and that a vaccine could go a long way to actually solving some of those issues. Is that right?
I believe it's a necessary step. I mean, it's not perfect in itself. I mean, access is still going to be an issue, so a vaccine would be great if it was equitably administered or made available. So there's not, the issues that we're seeing with COVID where rich countries are hoarding it. If it was distributed evenly and people that need it the most were able to get it, if it was made affordable. One thing that could pose a problem is, I'm learning from the COVID crisis that we're in right now is that people are really, really, really reluctant to take something that they need multiple doses of. And currently, just because of the nature of how hard it is to elicit the antibodies that neutralize HIV, all of the strategies that I'm aware of take multiple doses, so we call it sequential vaccination. And so that in itself could present some problems in terms of reaching people and making sure they're able to get the full regimen. So it's definitely something that will need to be tweaked even when a vaccine is available. But I think I'm very hopeful. I think prevention is always a better option than treatment when you're talking about a disease and a population from a cost standpoint, from a feasibility standpoint. I mean, I think it's the best shot we have. No pun intended.
And what do you think about folks who, the best shot, okay. I missed it, but that was brilliant. That's great. Oh, that made me happy. Okay. But what about folks like we're seeing across the US? There are a lot of folks who aren't sure about the COVID vaccine, for example. I guess, what do we call it? Vaccine hesitancy. Folks who don't think they really want to go out and get the COVID vaccine. How do we, if we were to develop an HIV vaccine, I could imagine that hesitancy would still be there. How do you go about reaching people to share the good science?
Yeah, so that's a really difficult question. I guess I'll start by saying a community that's not seen can't be served. So when we use the term vaccine hesitancy to largely characterize a group that typically, refers to people of color, the rhetoric that I'm hearing about that is that this group is opposed to getting the vaccine because of largely it's framed in reference to the Tuskegee experiment, which was a study conducted over half a century ago. And that's pointed to as the sole reason why people don't want to get a vaccine. And I think that's a really warped perspective for a lot of reasons. It's first, it's treating everyone like a monolith, as if everyone has the same reason for not wanting to get a vaccine. Two, I think that it dismisses legitimate concerns. So yes, Tuskegee was one instance in which the medical system acted unethically and bred a lot of distrust, but that was one incident. That history extends way before Tuskegee and persists through today. I mean, I personally have experienced this as recently as last week. So, I mean, it's something to attribute vaccine hesitancy solely to Tuskegee overlooks these constant interactions that are played with disrespect that African Americans experience on a daily basis. And so if you don't acknowledge that those things are happening, then you can't repair them. You can't begin to rebuild that trust. And so I think we have to be realistic with ourselves and not use Tuskegee as a scapegoat, but actually look at how the medical system or medical practitioners interact with people of color. I think vaccine hesitancy, using it in the context of COVID, people tend to use that as a wide paintbrush, the broad stroke paintbrush, and say people are vaccine and equate it with anti-vax. And I don't think that's the case. I think that one, there are populations that have histories that make them, they give them legitimate reasons to be cautious. Two, the COVID vaccine was developed in a very atypical way. It was developed much faster, and a lot of it hasn't been explained in ways that are accessible to people. And so, that can be scary. And I don't think that's irrational. I don't think that means that they're against vaccines in general, but I think that this is a different instance. And I think that these people are getting lumped in with the people who are like not getting the vaccine because they think there's going to be a microchip implanted. I don't think everyone is not getting the vaccine because of irrational fears. I know a lot of highly educated people who are also opting not to get the vaccine, not because of, historic mistreatment, even though that probably feeds into it, but just because they want more data. It's been a very short time since we've had this available, and they're in positions where they're not at high risk for exposure. And so they're just kind of sitting back to wait and see what happens. And I'm not encouraging that, but I can empathize with that frame of thinking. I can understand why people are nervous, or I would say cautious. This is something that's going into your body. And I would encourage anyone who, whether you're pro-vaccine, anti-whatever, to do research, like I guess my boss always says, do what it will take to convince yourself. I would encourage everyone to take the steps of like looking for credible resources and getting the data or, the information you need to support your decision, whatever that decision may be. I definitely hope it's to get the vaccination, but if it's not, you have to consider the context in which you exist and live. I mean, is the vaccine a good option for you? Do you have conditions that might make it not, that make the cons outweigh the pros of getting the vaccine? I think everyone needs to do that independent assessment of pros versus cons and decide. So I think that when we talk about vaccine dissemination at large, people of color and marginalized groups don't get that level of humanity when we're having those discussions. We're not afforded the credibility that we can think for ourselves and make decisions independently. And just because the decisions we make don't look like the mainstream, that they're ill-informed. Some of them, there are a lot of people who aren't well informed and are making the decisions based off misinformation. And I think that I do a lot of outreach work to try to target those people. But I think going into those settings with the mindset that we have to change people's minds and tell them what to do, it's just a terrible approach. And so even what I think thinking about things the way that I've heard, like the rhetoric I was describing, I think it leads to poor design of interventions for reaching those communities as well. Even though they're well intended, if you're thinking about people as a monolith or as, incapable of comprehending the situation at hand, then I think that you're definitely going to have a difficult time reaching them, and let alone changing their views. So it's a really important issue. It's a really difficult problem. I don't know the answer, but I definitely know that as a person of color working in science, who shares these experiences, but also sees the other side of what happens behind the curtain, that whenever I can, I try to use my role to kind of bridge the gap or serve as a liaison. So, to be a trusted face, to address those concerns, to answer questions on a level that people can identify with or understand, no matter how basic, in a setting where they're not intimidated or felt, that they're spoken to in a way that's condescending. I think that rebuilding the trust will kind of require that they see faces that they can trust as well. And so that's part of the work that I try to incorporate with my research as well.
I'm glad that you are. Brilliant. All I could do after that is just like round of applause and say thank you for coming on this program to share with us about the work that you're doing at Duke and the work that you're doing in the community. I think I actually saw online that you were working on some multimedia projects too with the Museum of Life and Science around some of this. So I'm excited to see what more you'll be working on in the future.
Oh yeah, absolutely. The exhibit, I think, will be released on Wednesday. So yeah. And there's some other cool projects in the pipeline as well.
Oh, fascinating.
Yeah.
Well, we will all keep an eye out on the projects that you've got going on. Christine, thank you so much. Absolutely just brilliant stuff. Folks, wherever you're at tuning in, it's time for audience Q&A. So be dropping your comments and your thoughts into the chat. I'm going to be turning to those. The moderators are standing by. They're sending me your questions. And Christine, I think you've probably got the answers. So the one that I've got right here, right now, in front of me is Glenn wants to know if you see mRNA becoming more important in the future of vaccine development.
Absolutely. From a vaccine design standpoint, so there's actually a pandemic preparedness plan that's the crux of it is using mRNA, just because it's so much faster to develop a vaccine rather than using proteins or DNA. I'm using a protein approach right now for my project, but hope to transition to mRNA. With proteins, there's so many things you have to consider, like how they fold and if they're floppy and all this stuff. Whereas with mRNA, you just give your body the IKEA instructions and it assembles it itself. You just inject it and it does its thing, and you don't have to worry about all of those issues of protein stability. So definitely, and it's just fast. I mean, with the variants, we're developing boosters that all you have to do is literally change the sequence. So kind of like you do an update on your phone instead of getting a whole new phone. It's like a software update. So it's a much easier platform to design vaccines off of. So yeah, and I think it's cheaper too.
All right, there you go. Next one here. How close are scientists to developing a completed HIV vaccine? Do you have an estimate of time?
I'm not a betting woman, so I'm not gonna say a time, but I know there's a lot of news surrounding. There's a researcher at Scripps, William Sheaf, and he has promising research out that they've got a candidate that might move forward. And there's lots of approaches that show a lot of promise. I think I don't know. I think there's still optimization that'll have to be done. A lot of things that we find in the lab that work in animals don't necessarily translate into people well. So I think we're getting closer, but I think it'll still be some time before we get there.
All right. Next question is from Danielle. What steps should the medical community take to bridge the gap in the communities that are afraid of the COVID vaccine?
Ooh, making the people of color. Wow.
Ooh.
Yeah. A simple step: representation. I don't have the answer to this. I don't think anyone does, but I definitely think representation. I think having it be a place where different cultures and ethnicities and stuff are represented at proportions that are consistent with their numbers in the population at large would be encouraging. Right now, that the people that are practitioners don't necessarily reflect the populations they serve, and I think that right there creates a disconnect in terms of even empathy, but definitely cultural competency. And so I think that making it a more diverse setting would be one start. I think that the communication. So this may be more of a US type thing, but how we interface with patients, I'm not a practitioner, but how practitioners interface with patients, like the doctor themselves, it's very clear in those interactions that there's a time limit. And so that makes a very unnatural setting to have a conversation. It's very uncomfortable. If you're a patient, you're coming in, you may not be knowledgeable about what the issue that you're having, or not comfortable or confident in your ability to address your concerns. But if you feel that you're on a timer and that the doctor's like, okay, I got 5 minutes, 4 minutes, 45 seconds, get it out and leave, that's just not a setting that's going to foster a bidirectional communication between practitioners and healthcare settings. So until we have a setting that's more comfortable and conducive for communication and building relationships, I think that there's always going to be some degree of mistrust. You have to actively work to repair those relationships. So I think how doctors interact with patients, how they talk to them. I mean, there's a lot of research to support that there's different beliefs about the pain tolerance that certain minority groups have, and that affects like when they go for treatment, whether their concerns are actually acknowledged or whether they're sent away or deemed pill seekers. I mean, there's so many issues that need to be addressed to improve how care is administered. But yeah, I definitely think more training and awareness. I think that it should be mandatory that practitioners are made aware of some of the ways that they're perpetuating these racist or discriminatory practices. And honesty, I think acknowledging it. I hate being in a situation where you can clearly see that something is happening and you have to convince the other person that they're doing it. I think that it puts the burden on the patient or the victim to fix the problem. And I think that's not the way to go. So I don't know.
That sounded like a fantastic answer. I think you do know. I think that was great. And a little bit earlier in the show, Danielle too had wrote, the medical community must rebuild trust. We need more people that, all caps, look like us. So I think y'all were thinking on the same lines.
Absolutely.
All right.
Representation.
Let's see. All right. Next one comes from Kevin Daniels. Will some of the new knowledge that scientists gained creating the COVID-19 vaccine be able to be applied to your current HIV research?
Oh, absolutely. Like seeing how I'm already trying to transition my project from protein-based vaccine to mRNA-based vaccine. So, I mean, yeah, definitely. Definitely. I think that they both inform, HIV informs COVID and COVID likewise is informing HIV. So, yeah.
Did the volume of like published research papers that you have to sift through now just grow exponentially once all the COVID papers started coming out?
I think I have one of my big notebooks with me now. Absolutely. Well, that's just one. But a lot of the things with COVID are preprints, and so they're not published yet. And so, yeah, it's a lot. I do my best.
Right. That's the one thing that I think about being a postdoctoral scientist, or what do we say, early career researchers. So you're not like all the way like academic professor scientist yet. It's postdoctoral. I don't have a really great definition for that one, but I feel like the postdocs and the postdoctoral researchers that I've met working at the Museum of Natural Sciences, because we have academic labs and research labs where people are working, they've always got like 75 browser tabs and 74 of them are research papers, and one of them is their email inbox. And then they've just got stacks of research that they're always pulling from. So that you reached down and you were like, yeah, here it is. I was like,
so bad. It's so bad. My poor computer, it's actually fairly new, and it sounds like it has asthma sometimes. And so I'll like restart it and I'll close out all the browser windows. And then I happen to discover that if you go to history, you can restore all the tabs. So I'm just like, of course I want to restore all 189 tabs. Of course. And so it's just like, it never really gets a break. Yeah, it's a lifestyle.
It's a lifestyle. But if we're going to develop a vaccine for COVID or HIV, or save all the lemurs in Madagascar, or find out what's causing chytrid fungus in frogs and amphibians, then that's part of it. Doing the research, making it available, and then crucially and importantly, doing this part too, I think, which is finding a way to get what you're doing out to the public, to the people in the places where it matters.
Absolutely. And I mean, I don't think papers would constitute making it available, since you have to have like an affiliation with the university to access them or pay hefty subscription fees or things like that. So I think this is how you make the research accessible. by doing outreach work and stuff like that. So yeah, definitely important. Taxpayers fund the research, so they have a right to know what's going on.
Absolutely. Well, everybody tuning in, you can go ahead and start dropping those clapping hands emojis into the chat box for Dr. Christine Daniels. Christine, thank you so much for being our guest on Science Tonight.
Thank you so much for having me. This is a lot of fun.
And everybody, thanks for tuning in to the show this evening. Hope that you enjoyed it. I know I did. This was an amazing conversation. Know that you can come back to this same link if you want to share this with your friends, families on social media. Copy paste this YouTube link, bookmark it, keep it handy, and share the show with everybody you know. We're here every Thursday night, 7 o'clock Eastern, right here on the museum's YouTube channel. Of course, you can also just click the subscribe button that's below the video there and the bell icon to get notified. That'll let you know when we're bringing you another great video out of the Museum of Natural Sciences. We publish great stuff all the time. And if you like this show, you'll probably like some of the other stuff that comes out of the museum as well. Make sure that you visit naturalsciences.org to see what's coming up in the future. And you can follow the museum on social media. We're at Natural Sciences on Facebook, Twitter, and Instagram. Next week on the show, we're going to be talking about evolution, exercise, and diet. That's right. Dr. Herman Poncer has a new book out called Burn, and it's all about the evolution of human physiology, how we burn calories, stay fit, and lose weight. So we're going to get the real science behind counting calories next Thursday night from Dr. Herman Poncer. He's done some incredible research looking at hunter-gatherer peoples and modern humans and food and where we get food and what food is made of these days. I'm excited about it. We're talking all about his new book, Burn, naturalsciences.org. That's where you get all the information for next week, including the link. Or, like I said, just subscribe right here to the museum's YouTube channel. We do cool stuff. Just hang out here with us, all right? So, hey, everybody, I'm Chris Smith, and I hope that you will take care, stay safe, and keep your community safe. I'll see you next week. Good night, everybody.